Welcome to today's lecture. Today's topic is marketing of liquid milk, or we can say the market milk processing. So this liquid milk is one of the most important part of milk processing and marketing, especially in India. Much of the milk is consumed as liquid. So earlier I have discussed about the milk collection or testing, reception, different kind of tests, transportation, etc. Now in continuation to that, today I will discuss the whole process of liquid milk marketing, but some of the steps will be discussed separately in more details in separate lectures. As I have mentioned, here we can see the milk consumption pattern in India. Almost 46 to 50% of milk in India is consumed as liquid milk, and that's what we call as market milk. As you know, the total milk production in India is around 210 million metric ton. The remaining 50% or 54%, that is ghee, is half of that, that is around 28%. Then butter 6%, yogurt 7%, condensed milk 6%, skim milk powder 4%, and remaining is paneer 2% and others. So this is a milk consumption pattern in India for an understanding about how the milk is consumed in India. Here we will see the flow of operation in milk marketing or liquid milk marketing. First is the receiving of milk. This part I have discussed earlier. So there will be sampling, grading, weighing and testing. So to some extent I am again going to discuss some other part of it. Then there is preheating. So milk has to be heated partially so that it can be further processed, mainly for filtration or clarification. So this is to remove the dirts coming with the milk, different kind of dirts or the feed material, etc. extraneous matters can come, so that has to be removed. That is by normal filtration or by mechanical way, that is called clarification, which we are going to discuss more details. And then after that, cooling and storage. So after this filtration, temporarily milk will be cooled for immediate protection. Later on only this milk will go for further processing like pasteurization, etc., which we are going to see next. So now the milk will undergo standardization. This is about fixing some specific requirement as per legal requirement, the fat content and the SNF content. So this is first calculated and then the proper mixing is done to get the exact level of fat and SNF or other legal requirements. After that it undergoes pasteurization. So that is a process of preservation, we can say. It is mainly to make the milk safe, to kill all pathogens, and partially it helps in preservation also. So we are going to discuss a full lecture on this topic with all details. And after that there will be bottling or packaging. So old practice was packaging in the bottle. Nowadays the packaging is done in the flexible plastic pouches by manually in small scale operations or in a automatic machines, that is called form fill seal, which we are going to discuss later. And after that it will go for storage. Sometime after pasteurization the milk will go to storage tank and chill there, and from there it will come for packaging and then storage and distribution. So this is the flow of operation for market milk distribution or liquid milk marketing, which we are going to discuss slowly step by step. So first we will see the milk reception operations. So this operation of receiving milk may be subdivided into several steps. First is like unloading. The milk comes in the milk carrying transport vehicles, which I have discussed earlier. Then there will be grading. So first there will be a platform test and whether it should be accepted or rejected, and then there will be further sampling for other tests. Then there will be weighing and then other laboratory tests. So these are the steps in the whole process of milk reception. before it goes for further processing in the pasteurization, etc. As I said, first is the platform test. So this is the test which is done at the reception platform. So this includes all those tests which are performed to check the quality of the incoming milk on the receiving platform so as to make a quick decision regarding its acceptance and rejection. So these tests are done very quick for immediate grading of the milk, and they are performed on each can or tanker of milk with the object of detecting milk of inferior or doubtful quality so as to prevent it being mixed with high grade milk. So at the preliminary stage we must understand the quality of milk, whether it is suitable for processing and whether it is suitable for accepting. Then only it can be used to mix with the other quality of milk for further processing. So platform test is very important, which we are going to see later. Now these platform tests are two kind. One is called rapid platform test, which is done very quickly and immediately on the platform, and some of them are other category, that is the other platform test, which takes some time. So in this rapid platform test, which are used mainly for the organoleptic or sensory test, which take very little time to perform. So in this, first is the smell. So when a milk can comes, we need to open the lid and immediately we will take a smell. If the milk is very old or if there is a lot of acidity, then we will get a typical sour odor. Then next is the appearance. By looking at the milk, we will get an idea. And then the temperature, we will get an idea by taking the thermometer. And sometime we can take a sip of milk in the mouth, in which we will get an idea about the sourness or even other smell. Sometimes there is a sediment test, which is kept in a small sample bottle or beaker, and very quickly we can understand if there is a sediment. If there is a lot of manipulation in the milk, there will be a sediment which can be assessed very quickly. Here are other platform tests like acidity. This is done by titration. So this tells us about the length of time the milk is hold, or if there is an iteration or acidity. This can be done even by measuring the pH. So this helps in acceptance and rejection of milk. This is applied as a quality control and also as a platform test. Then there is ethanol test or alcohol test. This is to determine the heat stability of milk, because milk has to undergo a heat processing. So if there is a lot of acidity, it will not be able to withstand the heat processing. So by this alcohol test, we can understand the heat stability. This is also considered as other category of platform test. And there is alcohol alizarin test. This is to determine both heat stability and pH of milk. This is also useful as a platform test. Here are other platform tests in continuation, that is clot on boiling. So this is again a very important test which can be done as a platform test to determine the heat stability of milk. So if there is more acidity, if we just heat, it will be clotted. Similarly, there are dye reduction tests. So they are either MBRT or Risajurin test, that is methylene blue reduction test. So this is to determine the extent of bacterial contamination and growth in milk. So it is indirectly gives an idea about the bacterial load in a short time. So sometime it is, in case of Risajurin test, it is very quick in 2 minutes. So that is called 2-minute Risajurin test is applied as a platform test, whereas in case of MBRT it takes little more time. And there is lactometer reading, so which gives a quick idea about the adulteration of milk, especially if the water is added. So that is also used as a platform test. Now the next step is weighing. Once the sampling and testing is done, milk is accepted, it has to undergo weighing, because this is very important for milk receipts and disposals and for making the payments of milk. So by weighing we should know how much milk is received, total quantity of milk, and also it is important for payments. So it is done by using a weighing platform for milk cans, or in case of milk tankers it is done by using the weigh bridge in a large scale. So you can see in the diagram there is a flow of operation, that milk comes, then testing is done, sampling is done, after that milk is received, then it undergoes the weighing, and then it will be going to the initial chilling tank, and from there we will get an idea about the total quantity of milk after weighing, and the payment is done. So this is the routine operation in the reception section. So here we will learn little more details about the sampling and testing, though I have already mentioned the platform test and there are other tests also. So the sampling is very important, for which certain basic procedure has to be followed. All these details will be discussed in our practical class. So it needs to be thoroughly mixed before taking the sample so that it really represents the actual milk. So asepsis should be maintained for bacteriological sample. When we want to do any bacteriological testing, the asepsis, that is the container or the sampling collecting devices, all the process should be totally free from bacteria. So that is the asepsis, free from bacteria. We should not add any bacteria in the process of testing. Apart from initially accepted, rejected lots of milk, there are always some of doubtful quality. So though we have accepted or rejected, there will be some doubts. So that can be further decided by further laboratory test. So milk is always tested in a quality control lab. So here, in addition to the platform test, some more detailed tests are done in the laboratory, which we are going to discuss in the next two slides. So here are the other laboratory tests for milk. Number one is MBRT, which I have already mentioned, that is methylene blue reduction test. This is mainly to determine the indirect microbial load. That is a laboratory test. It takes more time, up to 7-8 hours. Then there is standard plate count. This is a proper microbiological method for enumeration of the microbial load. So this takes more than 24 hours. This is a laboratory test. And then there is DMC, direct microscopic count. So this is to identify the types of microorganisms present in milk. So this is also a laboratory test. Then there is freezing point, or sometimes freezing point depression. So when there is a more adulteration, particularly by adding water, the freezing point will go down. So that is called freezing point depression. It will go below the normal freezing point. So to detect the adulteration with milk, we can go for freezing point test. And there is a fat and SNF test. So this is again very important for payment purpose. So fat percent and SNF, so which will be done details in the practical class by Gerber centrifuge method. So this is also a laboratory test. Now cooling of raw milk. Once the all tests are done, platform test and other tests, and satisfied and the milk is accepted, it has to be cooled and stored immediately. Otherwise, there will be deterioration and spoilage. But sometime if we go directly for processing, then it need not be. But generally the practice is, after it is accepted, it will go for immediate cooling and storage. And later on it will undergo for further preheating, processing or pasteurization, and then again cooling and storage for packing. So in case of milk, when it is accepted or received in the plant, it has to be chilled at 5 degrees Celsius or below and stored in cool condition. So to prevent deterioration, it is bacteriological quality during the interim period. So methods for cooling of raw milk is by surface coolers. So there are some surface cooling mechanism in which the chilled water will be sprayed, or there are plate cooler where the plate heat exchangers are used that will chill the milk by using the chilled water. And there can be sometime internal tubular cooler. So there will be two different tubes. In one tube the milk will flow, and the other tube the cooling water will be flowing. Sometimes there is jacketed vat tank in which the batch wise the milk is chilled with a jacketed tank. Storage of raw milk. So the milk which is accepted and chilled, it has to be temporarily stored until it undergoes for processing or pasteurization. So normally the milk storage capacity is equal to 1 day's intake. So how much milk we are receiving daily, that much storage capacity we should have. This allows uniform work for processing and packaging operations with less dependence on the time for receiving raw milk. So there has to be a synchronization, how much milk we are receiving and how much milk we are processing. There has to be a balance. The storage tanks, sometimes they are also called silo, are used in milk plants for the storage of raw milk or even pasteurized milk or processed products, often in very large volumes. So depending on the capacity of processing daily, we must have different kind of tanks either for storage of raw milk or storage of pasteurized milk. Now the next step is preheating. So once the milk is accepted and all the tests are done, the milk may be chilled and stored initially. After that, when we want to go for processing, the milk should be first undergoing the preheating. So preheating is a preheating before the other actual processing. The temperature will be 35 to 40 degrees Celsius. So actual processing starts with the clarification or the homogenization. For both this process, the milk should be little bit warm, even up to 40-50 degrees Celsius. Then only this clarification or homogenization will be effective. So as the temperature increases, the viscosity will decrease. So it will be more efficient for filtration or clarification or for homogenization. So for this heating we need to do in case of small scale, we can do it in batch heating process, but in a large scale commercial it is done by plate heat exchanger. So plate heat exchanger is the main system in which the milk is heated with the help of hot water or steam. There are thin plates arranged together in large number. So in one side the milk will be flowing and the other side the heating medium, either the steam or hot water will be flowing. So there will be heat exchange between these two, but they will never mix together because they will be separated by thin plates. So all these details we are going to discuss in separate lecture when we will discuss about the pasteurization. Now next is the filtration. As I mentioned, after preheating, first thing is filtration. This is to remove the external materials or dust, to improve the aesthetic quality of milk by removing visible foreign matter which is unsightly and may therefore cause consumer complaints. So filtration removes suspended foreign particles by the straining process. So this filtration can be done by using the cloth or by metal mesh also. So this cloth or the metal mesh can be fixed with a proper container and through which the filtration of the milk can be done. Then this makes the milk ready for the next step of processing, that is clarification. Now we will discuss about clarification. Clarification is a mechanical method for removing the foreign matters or dirt particles by using the centrifugal techniques. Like we use cream separator where the cream and the skim milk is separated by the centrifugal force. Here also by using the centrifugal force, the floating materials or dirt materials or particles can be removed. So this works with the force of centrifugation. Now there is location of the clarifier in the processing line. So it can be located in three different places as per our requirement. One is between reception and storage tank. So in that case it is not the warm milk, it is cold milk. It can be between regeneration and heating section of HTST. So this all we are going to discuss in details in the HTST pasteurization, and it can be either in between the preheater and pasteurizer. So as I said, there is preheating, after that pasteurization in the actual continuous pasteurization method. So there the milk is warm. So here we can see a comparison between the clarifier and the cream separator, or clarification and cream separation. So clarification is only to remove the non-milk particles, extraneous matters. And in case of cream separator, here the cream is separated from the milk. But both the cases the principle of gravity is used, or the density of the particles is the main factor. The centrifugal force is applied in both the case. But in case of clarification, there is only one outlet through which the dirt or particles will come out. Whereas in case of cream separation, there will be two outlets in which the cream, being the low density, will be separated and coming out through one outlet, and the skim milk being heavier or more density, it comes out through another outlet. But this separation happens with the help of centrifugal force. Here we can see the diagram and actual machine of centrifuge, cream separator and the clarifier. In the top right we are seeing the actual clarifier. In the bottom there is a diagram of cream separator, and in the left side also we are seeing a schematic diagram of cream separator. So in case of cream separator, the milk comes from the center and it flows down. There are hat shaped several discs, so those discs are packed. Now due to the rotation in high speed, a high centrifugal force is created which separates the low density cream to the periphery, I mean towards the center, and the high density scheme towards the periphery. After that they get accumulated and get separated through two different outlets. So this is the schematic diagram also presented in bottom right, and in the top we can see the clarifier. Here is another operation called Bactofugation. So this is a process in which the bacteria is removed, especially the spores. So here again the centrifugal force is used to remove the bacteria. That's why it is called Bactofugation. So it is a non-thermal process of removing almost 90%, 99% of bacteria present in raw milk, especially designed high-speed centrifugal clarifier. This is also a clarifier specially to remove the bacteria. So here the speed required very high, the centrifugal force required is very high. So two centrifugal clarifiers are arranged in a series, the first one operating at high velocity, that is 20,000 rpm. So here the primary objective is to remove the heat resistant spores which may not be killed during the pasteurization because that is not a very high temperature. So this is necessary to improve the quality, especially for cheese processing of milk. In the right side diagram we can see the machine for bactofugation. So in continuation about bactofugation, the bacterial spores being heavier than milk are expelled towards the periphery of the bowl. So bacterial spores are more density, so they will be pushed towards the periphery due to the centrifugal force. The bacterially clarified milk, then it is little bit lighter than the spore, so it goes towards the center and then they are discharged through an axial outlet. The bactofugation cannot remove all pathogens, so the process is supplementary to pasteurization, not the substitution of pasteurization. That means it can help in removing some of the pathogens or spores, but it is not a substitute of pasteurization. Pasteurization has to be done even after the bactofugation. Now the next step is standardization. After the preheating, milk will undergo clarification, sometime cream separation or bactofugation. After that we can go for standardization before actual pasteurization. So here it is a process of adjusting the level of fat and SNF as per the legal requirements or other requirements. So this is a process. First it is done by calculation. First we should know the fat and SNF in the milk, and then if we want to increase we need to add some cream or milk with higher fat. If we want to reduce the fat content in the milk, then we can add the milk with lower fat or skim milk. So this is first assessed, the fat percent we have to assess, and after that we have to make a calculation, and then accordingly we have to mix the lower fat content milk or skim milk to increase or decrease the fat or SNF. So this is further details will be discussed in practical class as a numerical problem. Now the next step is actual processing or pasteurization. This is a very big topic which will be discussed in more details in a separate lecture, maybe one lecture minimum. In details we will discuss about the pasteurization. So basically here milk is heated first, then it is cooled down, and then it is stored, and then it goes for packaging and marketing. So here in the red section you can see that is the heating section, then the blue section is for cooling section. So milk which is already there chilled after the reception, first it will come here, it will undergo heat treatment. There are different techniques, by batch method or by continuous method. In continuous method the plate heat exchangers are used, as you can see in the right side diagram. There are 3-4 different sections. The first section is regeneration, then there is heating section, and then there is cooling section. So all these details we are going to discuss. There are different temperature, either low temperature, it is around 62 degrees Celsius for 30 minutes. There are high temperature and short time, which is 72 degrees Celsius for 15 seconds. And sometimes there is ultra high temperature. So all ultra high temperature separately we are going to discuss in another lecture. So all these things we will see and discuss later. Now the next step is the packaging. So after pasteurization, milk will be chilled and stored again in separate storage tank. From there it will come for packaging. So the earlier days the packaging was in the bottle. So for which there are different setup of plants in which the milk will be filled into the bottle and then it will be sealed in case of large scale, whereas in case of small scale it will be done in a small plant where the milk will be filled and manually it will be sealed, whereas nowadays in big dairy plant it is done very continuously automatically by using the packaging machine. So this machine works as a form fill seal. So in the diagram we can see the plastic pouch. The plastic roll will be there in the other side, back side of the machine, that comes from that side, as you can see. Then it get sealed first vertically and it makes a pouch. After that milk is filled. The quantity of milk we can decide, it can be 250 ml or 50 ml to 500 ml generally. Now after the milk is pouring, then there will be sealing and that makes one pouch ready and starts for another pouch. So this is the technique for form fill seal. As you can see, the plastic pouch, it can be of different category. Now we are at the end of today's lecture. In some previous lecture, I have already discussed about the milk collection or reception and basic testing and transportation and other aspect, clean milk production. Today our topic is the understanding about the marketing of liquid milk. So we have discussed the flow of operation and some of the important steps like testing, platform test, other laboratory test, and then there is preheating, clarifications, or cream separation or Bactofugation, but some of them separately we are going to discuss, like cream separation separately we will discuss, and then after that there is actual standardization followed by pasteurization. So pasteurization again is a details separate lecture we will discuss. After that the packaging and then the milk goes for marketing. So in short, this is the flow of operation for market milk or liquid milk marketing. Thank you.